director, and he's a great professor of genetics and genomics at the University of Pompeu Fabra. He has been the director of the Department of Experimental Sciences and Biomedicine too. He was former director of research in the government of Catalonia. Secretary General. Secretary General. And he has more than 200 papers in high-impact journals, many of them, on genomics, different aspects of genomics and diseases. And he's an excellent speaker, and I remember the good days of that we were together at the Societat Catalana de Biologia, remember? Good times before the centenary.
That's right, that's right.
And I'm sure that you are going to enjoy very much his talk, which some of us already saw, but I thought when I saw it that it would be very appropriate for the ICM community, talking about the relationships between the genomes and phenomes, okay? So go ahead, and a pleasure.
Thanks very much, Francesc. Thanks very much for making me president and so on and so forth. And thanks for accepting me here today. I'll do my best, even though I have to tell you that when I prepared this talk, I prepared it for a general audience and not for people who, like you, work specifically in some of these topics, right? So I'm not so sure this is going to be a great talk by a great speaker, stuff like this. We'll see, we'll see. The leitmotif of my talk is going to be diverse, but I should start by saying something lest I forget, right? And the something is, part of what I will do are massings, philosophical massings that come from my mind. But another part of what I will present are real results by my team, who has been changing over the years, and to whom I owe pretty much everything that we are producing, right? And today I will perhaps mention stuff that Eva is doing, stuff that Jordi is doing, well, and others here. We'll have, thanks very much, we'll have a bit of a stuff about the scientific method, and this is, I'm afraid, a bit philosophical. Then I will tell you stuff about big data and genomics, and I will tell you about one project I'm involved in. And then we will mention stuff about genomes and phenomes, and we will get to biodiversity, right? And then I have a couple of examples. And depending on the time, we will do both examples, which are results from my lab, or perhaps just a single one, right? Oh my God. This is quite annoying, because of course, as usual, I forgot to cancel this, right? Now I have not forgotten. Okay, so let's go ahead. The scientific method. We all here try to do science, right? Or try to help science on. But what science? What is science? What separates science from pseudoscience? What separates what we do from what flat earthers do, from what climate change deniers do, from what anti-vaxxers do, right? What's the difference? Well, we have had lots of philosophy over the last 200 years, and there have been many advances on philosophy. But after 100 years of epistemology, the bit of philosophy of science that deals about what can we know, how do we know it, what science in a sense, we have learned two basic things. First, there's not a single demarcation criteria. Some of you might have read Popper or know about Popper. He was the one of my favorite philosophers. About 100 years ago, he came up with an idea, the idea of falsibility, right? Claiming that this was a demarcation criterion between science and actually metaphysics, right? But we know that by falsibility, we don't grasp everything that science is, right? There's not a single thing. There are many things that separate science from non-science. And actually, there are many different activities that constitute science. You want science to be testable, you want it to be falsified, you want it to be based on evidence, and so on and so forth, and you have a difference between astronomy and astrology, right? But actually, science is the conjunction of many different activities that include, of course, hypothesis generation, observation, comparison, experimentation, prediction, evaluation, and so on and so forth, right? Everything that I have in this list, and many other things, can be done with different proportions. Nowadays, for instance, we say in the era of genomics, right? And people are doing lots of description. And some people go and say, description, that's not real science. Good science is only hypothesis testing. And that's just not true, right? There have been other periods in history, the 17th and the 18th centuries, in which people gathered for 200 years many data. And then somebody, by looking at this data, could generate hypotheses and theories, like the theory of evolution, right? So we are in an oscillating field. And now we live through, in a sense, in which we are gathering data, right? And if you look at things from this perspective, you can do science in very different ways. You can do primary or secondary research, by secondary meaning use the data that other people have generated and published. You can do quantitative and qualitative science. You can do descriptive science, or you can try to do causal research, and so on and so forth. There's many different things that you can do, right? So let me clarify this, because what has happened is that entered the 21st century, we have gone again back to the 17th and 18th centuries, and we are gathering data. That's one of the main efforts, not only in biology, but also in many areas of science. And we all have heard about big data, right? What do all these things have in common? All these things are data that is available out there. You can go to YouTube, you can have your wearables and get your own data, you can have your Google Maps and your location, you can have your bank records, everything. All this is big data. And if you think for a second, why do we call it big data? What does it have it in special that we call it big? Think about it for a second, right? It has been estimated, there's the references here, there's several other references. These numbers change a bit, but not the orders of magnitude. It has been estimated that since the beginning of recorded history, humans generated 5 billion gigabytes of data until 2003. This is a lot of data, right? In 2011, the same amount was created every 2 days by the whole of the planet. In 2013, this same amount of data, 5 billion gigabytes, was created every 10 minutes. And of course, admittedly, many of these things are just cute videos of kitten in YouTube. And of course, you've got stupid photos that we make with our telephones. I left it there. And these telephones just record photos and send them to the cloud. And I was talking some days ago, a couple of months ago already, to some Apple executive telling me that the photos that we upload in our iCloud, we ourselves never look at again. So you can imagine big rooms of computer servers storing data, storing photos that we will never look at again in our lives, right? But also some data is interesting, right? Now, by 2020, we generated 1.7 megabytes every second, every person in the planet. And the estimate is that by 2025, there will be 175 zettabytes of data in the global data sphere. And if you wonder what's a zettabyte, it's a new word. We have heard about petabytes and exabytes, but of course they have had to invent zettabytes and yottabytes, because otherwise, there's a huge, humongous amount of data. Now, as I said, this data, it's not only useless information, there's plenty of very useful information. You've got pictures, videos, medical records, transaction vectors, GPS signals, and so on and so forth. This huge amount of data is big data, is what we are gathering now and what we are trying to make sense of. However, today we will not talk about all this data. What happens is that there's a fraction of this data, a small fraction of this data, that is relevant for human health. It's medical genomics data. It's a tiny fraction, that's sure, but it is indeed a huge amount of data nevertheless, right? I'm generating relevant medical data while I speak. Perhaps these stupid watch things that I'm walking, right? Or that I'm exercising or whatever. But perhaps it's not very good data, but it's nevertheless data, and it's data that can be used. These all started, and I love this slide, these all started more than 30 years ago. This slide is the presentation of the first draft of the human genome by of course, you have here Collins and Benter But of course, you might recognize as well Clinton and Blair, right? At the time, the President of the United States and the Prime Minister of Great Britain. I love this photo for two reasons, right? The first and the most simple photo, the reason is that this was the first teleconference ever done simultaneously in two different locations, right? And they were very proud about this. Now we can do it with FaceTime, with Zoom, whatever. We do it every day, many times. At the time, 22 years ago, this was a great thing. But there's another thing, more fundamental thing, that makes me love this photo. And this is, please, particularly the young people, think about it, right? Neither Clinton nor Blair were involved in the conception of the Human Genome Project. They were not there. What actually happened is that there was a bunch of people, including Watson, for instance, that went to the politicians of the time and sold them an idea that would involve investing a huge amount of money, $3 billion, in a project which would provide results after the politicians of the time had left office. It was a long-term investment. Clinton and Blair were perhaps getting votes out of decisions that had been taken by politicians from other political parties. And this is something that I should say, it's pretty unusual, but it is possible. So let's have hope in humanity, right? Now, these guys generated the first draft. And as I have said, the cost was $3 billion spent over more than 10 years. What people don't usually know, people kind of remember this figure, 3 billion, right? But what people don't know is that every year since 2001, the expenditure in genomics all over the planet has been larger every year than 3 billion. It was a bit over 3 billion for 10 years. Now it's probably more like 6 billion, 10 billion every year, right? That's a lot of money. Mostly in the UK and the United States, of course, but not only there, right? This generated a huge amount of data. All this money spent in genomics has generated enormous amount, lots of genomes, not only of different species, but many human genomes, expression data, single cell data, of course, array-based data, GWAS, right? And if you want to look at it, 2 years ago, exactly 2 years ago, it was the 20th anniversary of the presentation of the first draft of the human genome. There was these two special issues in Science and Nature. Please look at them, because you'll get an idea of the explosion of data and the enormous diversity of data that this human endeavor has produced. This kind of data gathering planet-wide project that we are doing, right? And this, I like this slide, but this was already in the 10th anniversary, not even the 20th. Now, they cannot do this slide because, of course, it's just so big, so large. You have many different, you have the ENCODE, you have GTEx, you have the Earth Biogenome Project, you have many initiatives that deal with genomic data. Okay, this is big data, this is medical genomic data. Now, in terms of human health, over the last 15 years, the STAR method, the method of choice to link genomes and phenomes to solve this enormous mystery, very relevant mystery, has been the genome-wide association studies. And when I say phenome, and I say genome, I refer to these two words in their broadest sense, right? We're talking about genome variation, usually within single nucleotide polymorphisms. But of course, with a GWAS array nowadays, you can tag copy number variation. You can sometimes even study methylation. You can do many things in terms of genome variation. And also phenomes does not only mean disease status, does not only mean cases and controls of, I don't know, type 2 diabetes. It means every single phenome. I mean, for God's sake, there's even a GWAS with more than 1 million people about academic attainment, right? A good GWAS about your genomics to get this particularly interesting phenotype, having a PhD, right? And the results, by the way, are that there is some genetics, but it only explains about 1.27% of the variance. So most of the chance of you getting a PhD is not your genetics, it's how hard you work. So bear this in mind, right? Still, you know how it works. You get cases, normal samples, disease samples, you extract DNA, and you see if there are variants that are more frequent in the disease samples, in this case the purple variant, so they are the risk variants, or variants that are more frequent in the normal samples, and so they are protective sample variants, right? And then, of course, you present the results using Manhattan plots, and perhaps you have seen this because this, the first time I heard the word Manhattan plot, I had it explained to me, and of course now everybody knows that this, if you want positive results, this should look like the skyline of Manhattan because all these towers are skyscrapers that are very high, and those are your positive results, and the bits of the genome that are associated, for instance, in this case, with rheumatoid arthritis and so on and so forth, right? This has generated a humongous amount of results. I'm talking about thousands of papers, not hundreds, thousands, perhaps even tens of thousands of papers, and literally hundreds of thousands of genetic associations. This is the GWAS catalog, and every one of these dots, and this is just a synthesis, every one of these dots is a significant association. And when I looked at this for the last time, this was probably last December, it had almost 30,000 different studies from at least 500 different publications, right? This is huge. But we do have a problem with this data. Big data has a problem. We all have heard about privacy issues, right? We all have heard that all this data that we share, perhaps Facebook wants it to put it to bad use, essentially to sell us things, right? And we don't want our privacy to be violated, but we have a problem. Because of course, this is a human right, very clearly. Privacy is a human right. However, there is another human right, which is our right as humans to partake in science, not only to benefit from the results of science, but also to participate as citizens in science. Right? And all these medical genomics studies generate an issue. The issue being that, particularly when they have been generated using the taxpayer money, our taxes, if I'm a volunteer in one of these studies, I want that my donation, the donation of my blood, the donation of my information, is put to the best possible use, not only to produce a paper and that's it. I want this to be analyzed with new methods every time a new method appears. I want this, my data, to be part of a meta-analysis. And we all as citizens have the right to get these scientific results. So how do you balance privacy and the right to do our obligation as scientists to do the best possible science? How do we manage this? Well, there are several initiatives to solve this conundrum, and one of them is the European Genome Phenome Archive. The European Genome Phenome Archive is the response of the European Union to this problem. It's a big archive where we keep data safe, and we make sure that this data that is kept safe, and so no one can enter it and break habit on it or put it on Facebook or whatever, is, however, shared by shared with scientists all over the world. This European Genome Phenome Archive is run from here. I direct it at the CRG. I also work at CRG, but that's another issue. And the data are in the Mare Nostrum, in BSc, and also it's co-directed by our colleagues in Kingston in the European Bioinformatics Institute. And it's not big data, but it belongs to big data. At the moment, what we have in the BSc is 16 petabytes of data with more than 4 million files. We have 24,000 requesters all over the world to whom we distribute this data. This data comes from more than 4,000 studies. We have more than 9,000 data sets. We have more than 2,000 data access committees because each institution has one of them. So we manage all this. And the deal is we make science possible because, imagine, I want to do a meta-analysis and I want to gather 10 different studies from Japan, from Canada, from Australia, from Germany, from the United Kingdom, Spain, whatever, right? If I'm a single researcher, I should deal with 10 different institutions, perhaps in 10 different languages, with 10 different groups of lawyers. That would be hell. What you need to do now is just enter this single data point, EGA, and we will do most of the paperwork for you, right? Which is something I'm quite proud of. And indeed now, because We want to incorporate medical genomics data. We are turning EGA into a federation. It's not only 2 central nodes. There will be EGA Central. There is already EGA Central, but there are different local EGAs. And indeed, this started last fall. We now already have, sorry, I forgot Finland. Finland, Norway, Sweden is there. Germany and Spain are part of the European Genome Archive Federation. And actually, interestingly, Australia, South Africa, Canada, Argentina, and indeed, in a couple of weeks, I'm flying over to Mexico. They want to be part of this federation. So interestingly, this, managed from Barcelona, may become the largest operation in the world to make medical genomics data accessible to scientists everywhere, right? So this is quite interesting. But let's go back to, let me stop the publicity bit here. Let's go back to this kind of studies. Has this been successful? Perhaps some of you have had conversations with other people saying how all this genomics data, when will it get to the clinic, right? The answer is that it is already there on the clinic. Just look at all the drugs that were approved by the FDA by 2021. 2/3 of these drugs, of the approved drugs by the FDA in 2021, had genomic evidence supporting the usage of the drug. We already, if some of you, hopefully this is not the case, but if some of you has colon cancer or breast cancer, and you go now to the doctor, you will have your genetic profile done to make sure you get the right chemotherapy, right? And this is already getting the fruits of the 3 decades of genomics effort, which is quite good. So I could stop here because that's mostly what I have been working in over the last few years. But I promised to talk about biodiversity, right? And the thing is that most of the money we have spent in genoming at whatever level, humans, which is okay. But that's because that's because we were having not totally correct ideas. Let me explain what I mean by that, right? As I said at the beginning of the talk, one of the main tools of the scientific method is comparison. You compare things. Scientists compare things all the time, cases and controls. In an experiment, you should have a control. If you want to know what the gene does, you knock it out and you compare the cell, for instance, with a working gene and without a working gene, and you see what happens. It sounds childish, it sounds simple, but it is what it is, right? You compare things. You compare temperatures and distributions of soil density, whatever, and by this, you learn, you hypothesize, you generate your theories, and finally, you reach knowledge, you understand, right? Now, the question here is that everything I have explained about GWAS, genome-wide association studies, essentially compares humans with a phenotype, say a disease, Crohn's disease, and humans without the phenotype, without Crohn's disease. And that's not bad, right? But let's think about us trying to learn some other thing. Let's imagine we are aliens, ETs, landing in Earth when humans have already become extinct, right? And we need to learn about architecture, for instance. How will we learn better architecture? By comparing homes that are very similar to each other. I don't know, an American suburb, for instance. Hey, we can compare this home with this other home, but they are essentially the same. The painting in the wall may be different, the wood in the door may be different, but the homes are essentially identical. Humans are a species that is very similar. It has very little genetic diversity when you compare it with, I don't know, chimpanzees, the species next door, right? And actually one can say, hey, let's get larger sample sizes. Okay, fine. But if you get larger sample sizes, essentially you have a whole lot of different American suburbs, right? And indeed, you can get humongous sample sizes, just as the large consortia that do human disease now by the millions, and still they are very similar houses, right? I pose that even though we might learn something about human phenotypes and about disease comparing things that are very similar to each other, houses in Badia del Vallès and houses in L'Hospitalet de Llobregat, we perhaps can learn about luminosis here, right? Not a problem. But we will learn much architecture if we compare many different kinds of buildings from many different cultures, right? If we compare Roman architecture with Maasai architecture, with Hobbit architecture, with New York, and actually with my hometown, Nalek, this is my room, by the way, we may learn more. We will see different pipes, different tricks to avoid the damage of rain. We will see different kinds of ways to hold a building together with very thick walls or with just, steel and glass. We will learn these things. And so, rather than looking at humans to study phenotypes, because humans are all the same when you look at them from a distance, we should look at very different species that differ a lot within these phenotypes, right? Thing is here, let's assume I'm interested in studying bone density. Well, of course, I can look at different humans, but why don't I look at different species that do have very different bone densities, right? This kind of approach came to our minds when a few years ago, already 5 years ago, we did a study. And the study was about longevity. So longevity is a very important problem. We age and we have senescence. I mean, you look at me now, of course, and you see, hey, why this guy is talking about senescence? This guy is very young, and indeed he looks like Sean Connery, let's face it, right? Well, of course, Sean Connery in The Name of the Rose, not Sean Connery in James Bond, but that's another matter. But of course, we can keep comparing me with other people who have aged less gracefully, Xavier, for instance. But indeed, why don't we compare mammals, right? Mammals have a huge variability of aging. Indeed, you've got mammals that live almost 200 years. It says 95 here, but actually it's between 1 year and 200 years, right? You have 2 orders of magnitude of difference in mammals. And why do we age, we were thinking at the time? We were not thinking about studying mammals. We were focusing in humans at the beginning. And what we did was a study in which we tackled the mystery of senescence, the mystery of the shocking fact that our organisms are able to produce from a zygote, from a single cell, organisms as complex as us here, but then we fail in the maintenance, right? And we lose our hair and we become wrinkled and stuff like this. What's going on, right? Well, what we realized is that there have been lots of research in many different species, but the 2 classical evolutionary theories had never been tested in humans. And there are, I cannot go into details with this. There were 2 theories, the mutation accumulation theory by Peter Medawar and the pleiotropic theory of senescence. And essentially what we did was to translate these 2 ideas, mutations that act when you are old and destroy you and are bad for you, natural selection doesn't see because you have already had children and grandchildren, right? And George Williams, actually gave a twist to that, and he said, hey, if these mutations happen to be good for you when you're young, if you have, I don't know, you absorb lots of calcium from your diet when you're young, this is good because your bones are stronger, and because if you're a mammal, you will breastfeed your offspring much better, right? You have healthier offspring. But if you accumulate so much calcium when you are old, you may have arteriosclerosis, right? But hey, that's not the problem of natural selection because this mutation has been favored. It's already in the next generations, right? So we translated this into human disease. And what we studied essentially was mutations that are protective mutations from early childhood diseases and that perhaps are risk mutations, the same particular mutation is a risk mutation from a late onset disease. And it doesn't really matter. We actually found out that the predictions of the mutation accumulation theory and of the antagonistic pleiotropic theory actually fit what you observe when you study all these GWAS data that we can obtain from the European Genome Phenome Archive. Let's forget about this paper. The most important thing here is that when we got a list of the genes that contain these particular mutations, the pleiotropic mutations, the same mutation having these 2 effects when you're young, when you're old, well, we said, hey, are those really genes that have to do with aging? And we compared with aging genes that have been obtained in other species. In mice, for instance, right? 135 genes that change their expression. It's not even mutations, it's gene expression levels between old mice and young mice. And many other studies, right? And it turns out, we realize that they are the same genes. The genes that we managed to link with aging using human disease data overlap very significantly with the genes that actually have been studying worms or mice or whatever species have been experimentally linked with aging in those other species. And that's actually what gave us the idea, hey, rather than studying senescence and aging only in humans, why don't we open our range of search and look at primates or mammals, because we have actually longevity data for these species, right? And that's why we started doing genome-phenome studies over phylogenies. Now, remember this slide, right? We're focusing here, but we had to change focus. Now, we have been focusing on here, for a few years now. And what are the results we find? Well, we published this a couple of years ago. This was our second attempt, actually. We used mammalian data. We used genomes, publicly available genomes, from 60 different mammals. And we classified them between long-lived, in yellow, including, of course, humans, and short-lived, right? And what we did was very simple. We looked for amino acid changes in protein coding genes that were shared by all these long-lived species, like everyone here has Y or R, and that were different from the short-lived species, right? And we ended up detecting a bunch of amino acid changes, over 1,000 of them, that follow this pattern. And of course, that's a statistical excess, which proved that this was a statistical excess. So we have a list of amino acid changes that are shared by all long-lived species, and notice, please, that they are all over the place. Bats, primates, whatever, whales, and then a bunch of amino acids that are shared between the short-lived species, right? And now, of course, we are following up this research line, and we are running experiments. We're running CRISPR experiments to see whether these mutations have an impact in cell cultures, and we are crossing the human with the mouse barrier and so on and so forth. The point here is that we didn't have to run an experiment to get this data. We just went to the public wealth of biodiversity data that other people have gathered, both genomic biodiversity data, but also data about the phenotypes of these species. And we gathered this data, we put it together, and we were able to pinpoint at a bunch of different mutations that, interestingly, we could not have seen by GWAS. Why? Because, think about it, right? We all humans are long-lived, much long-lived than our closer species. So perhaps, if we are studying longevity, perhaps the mutations that determine our phenotype, the most important ones, the ones that are the big columns that hold the building, were fixed perhaps 2 million years ago. So now we cannot see these mutations. We cannot pinpoint these genes only by comparing some humans with other humans, right? We cannot do that. GWAS is blind to the information that biodiversity produces. And now we are extending all this. We have obtained data for more than 200 species, primate species. We have obtained more than 200 high-quality phenotypes, and they are all over the place. They have to do with ecological traits, behavioral traits, morphology, physiology, life history, and this is the different measurements we have in stuff like, the morphology and physiology of the vestibular system in the ear and so on and so forth. We are running this study. We can compare correlations about different relevant traits like body mass index and many other traits. Sorry, not body mass index, adult body mass. I'm talking about primates, not humans. And we can see how biometabolites correlate with one another. I mean, we want to understand levels of cholesterol in humans. There are different species out there that have very different levels of cholesterol, right? We want to understand cancer in humans. There are primates that have much higher levels of cancer, and there are primates that have almost never have cancer. Why don't we do this comparison? rather than just limiting ourselves to compare humans. That's boring, for God's sake, right? Anyway, we can do all the correlations. We have all these data sets and so on and so forth that I have mentioned. Let me avoid the detail here. I'm just pointing out, of course, if you're interested, I can dwell on this. I'm just pointing out that this is really interesting and it's being illuminating. We already have some results. And indeed, because we have all this very complex phylogeny, we can track this particular red branch here, which is the branch that leads to humans. If you're interested in the evolutionary history of humans, you can learn what are the phenotypic changes that were actually happening in this branch. You only need to use classical evolutionary techniques, ancestral trait reconstruction. And actually, you can use also classical evolutionary techniques to find out what are the amino acid changes or whatever kind of change that you want to study that took place in these branches, right? We can track this particular branch. And we are getting results like this, right? It doesn't make too much sense to dwell on them, but we are detecting a bunch with two different methods we use, changes in rates of evolution and conversion amino acid substitutions. We are getting results that are genes that have to do with our biometabolites, with our body mass, with our brain morphology, with our diet, with our longevity, of course, with our sexual selection and behavior, and so on and so forth. Those genes that we are detecting had never, even though, of course, they are relevant for human health, they have never, ever been even considered by the GWAS studies. Of course, as a sanity check, we are also detecting some genes that we already knew about, right? Because of course, some of the genes that have these mutations fixed many time ago, I still have nowadays segregating variation, so they still are relevant for the phenotype today, right? So anyway, these are, I don't know, I could give you examples, like for instance, for social organization, con-specific violence, so the probability in which one species has fights, usually between males, but not only, within the species, this, and humans, you can imagine if we are top or bottom, you can imagine whether we are particularly violent amongst us or less. This has to do with TET2 and SHE2 genes, right? And I use the example because some people in the audience sometimes work with these two genes in other species, and that's kind of interesting. You have these two top hits over here. This is quite interesting. And also, there are other examples. For instance, I don't know, there are a bunch of other genes that have to do with visual acuity or microcephaly that we have detected when we have studied color vision or when we have studied cranial morphology, right? So anyway, the point here, the basic thing here is that we develop a method that allows to link traits over phylogenies, and that is complementary to what we have been able to do with studies that focus only in human phenotypes, right? And we are now studying many traits in primates. We are studying aging in mammals in a consortium, and also we are studying cancer in mammals in a consortium that still has no name, because we have managed to get all this cancer data from American zoos, right? And we hope that this is going to be complementary and that we will be able to help a lot, right? Now let me finish with a little coda, right? Some of you may be thinking, why is this relevant? And I like to explain that, of course, this idea of comparing species that are very different in their phenotypes is an idea I love, but it's certainly not mine, or at least not only mine, right? That other people have had this idea. And indeed, one person that had this kind of idea is Svante Pääbo. Svante Pääbo got the Nobel Prize last year, not because he managed to got DNA from Neanderthals only, not because he managed to get DNA from Egyptian mummies and stuff like this. The reason his discovery of ancient DNA techniques is relevant is because it widens the range of comparisons we can do. It opens a new field in science, because now, to study modern humans, we can study Denisovans, we can study Neanderthals, we can study extinct species. And so, we can do more and better science. That's why his discovery is relevant. And it's relevant for medicine and for physiology. Because again, to make medicine advance and to make physiology advance, we need to compare. And he has allowed us to compare with things that we couldn't even imagine 20 years ago. Right? That's, I would say, the final message. Comparing is fine. Let's compare more. If you want, if the comparative method works within humans, let's extend it to other species. And now, precisely, to be able to do that, it's time for us, for everyone that studies biodiversity at whatever level, to be aware of this and to include biodiversity in the priorities of human research. So just let's compare. And that's it. Thanks very much. There are mics over there, yeah.
Thank you very much for your nice talk. I have two questions. One is more philosophical and the other one more practical. The first one would be if you think that artificial intelligence could be the end of this gathering data era. And the second one is, okay, when you find the genes that overlap across a species, maybe you find some candidates that have different function or opposite function. So would they be still important for this phenotype, or because they act differently in a species-specific manner, they are not, or they are relevant for the process that you're studying? Thank you.
Okay, short answers and then long answers. So short are no and yes. And now long version. Thanks. Those are two very sophisticated questions, so it's going to take a while, right? So I do not think that artificial intelligence is going to end data gathering. I think data gathering is here to stay because essentially it has been automatized. You don't need to gather data, you don't need to hope into the beagle and spend 5 years of your life. Other methods are allowing us to gather data, and this still is in crescendo. Artificial intelligence may help us, on the other hand, to make sense out of this data. May help us to generate hypotheses, may help us to detect patterns, and actually, given ChatGPT, it may help us explaining our hypothesis to different audiences, right? Because perhaps they will even write the paper for you. So what perhaps will end is the era of the scientists generating hypotheses essentially alone. We can now think together with machines, and that's interesting. Second, yes, once you get a candidate, it's a candidate. So you cannot know for sure whether the function in this other species is going to be different, right? Indeed, with mammals and primates, which is the example I have used, one can hope that such similar organisms are such that genes have not changed function radically, right? But what you point is totally true. I mean, you point at something that people perhaps don't even know, right? A given mutation may have very different effects depending on the genetic context. That's very clear in experiments of artificial evolution in viral particles or in bacteria, right? These things evolve so fast and have so many different mutations that you look at the same mutation 1,000 generations afterwards and it does a completely different thing. Why? Because it interacts with a plethora of other mutations that have already changed, right? So yeah, these genes are just candidates, so one has to run experiments, right? There's one thing that we tried, I didn't explain this, right? We tried to see if the genes we detect for some phenotype, for instance, longevity, this particular, say, 1,000 genes, if they are enriched with GWAS hits. So if what you see now having an impact in the variation of the trait in humans is the same thing that seems to be relevant in the phylogeny. And the answer is no. And this suggests that there may be some decoupling between macroevolution and microevolution. It suggests that genes that are really relevant may have strong mutations and that these strong mutations, once fixed or lost, then they are already fixed or lost. And that what happens within a species, those are the details, and those are sorted out by other genes that are not so strong, right? Which means that if you want to alter the phenotype, perhaps you should look at other candidate genes that the phylogeny is illuminating, right? But of course, we cannot know. We have not proved this at all. That's why we're trying to run experiments with some selection of mutations and so on and so forth. Sorry, it took long, but those were two very good questions. What else do we have?
More questions?
So, thank you very much. It's a very interesting thought. So I have a couple of questions. One, if there is any criteria that we can use in order to choose which species we want to use to do this kind of comparison, and then if there is any area in which this comparison more success in doing this kind of comparison, I mean, cancer instead of aging instead of...
Totally. So, oh God, I have to, so I'll be totally honest, right? What we have done so far is not to select the species. We have used whatever we can. We cannot possibly pay for the sequencing of 200-something primate genomes. Why? Because first you have to get them. And some of these species are endangered species. You have to go to Africa, get DNA from feces. It's complex. So getting these genomes has been a huge investment by a large number of researchers. We couldn't have paid, so we have done what we can. However, your idea is correct. It would be ideal to select the right species. It would be ideal to get whatever, insects, plants. There are phenotypes out there that can be analyzed from the perspective of, I don't know, studying the cell cycle. What regulates the cell cycle? I mean, you can do this with cross-species comparisons, right? But we have not done that, and I wouldn't know how to start. The second part, yes, there are areas in which we know, when we think about health, there are areas that I think that are more relevant. I mean, hairiness, of course, amongst our phenotypes, it's hairiness, right? Because there are mammal species that have lots of hair, and there are mammal species that don't have hair, right? And I think it's relevant for some people in the audience, but I'm not too excited about this. However, I'm really very excited about the cancer data set. Actually, we already have done some preliminary studies there, and we're pinpointing, of course, if you do a cancer study, if we did a phylogenetic study with cancer and P53 did not appear, I would doubt our results. But of course, P53 appears, right, which is good. And then a bunch of other genes do appear as well. And that's quite interesting because I think that this area really holds promise. This combination of biodiversity and human health not only is very interesting scientifically, but also it's another argument if you have to discuss with a selfish person that doesn't like biodiversity, you perhaps can use the angle of, hey, perhaps you care about your health, right? So, thanks again. I don't know if I have answered what I have tried to. Thanks very much. Okay, good.
Thank you, Arcadi. Very interesting talk. I have sort of perhaps more philosophical question or comment regarding aging. No, aging, to me, it's something very, particularly when you think in terms of different species. No, aging is, and the concept of time probably is very different in different species. No, because what is your heart rate, or what is the heart rate of another mammal, no? Thinking in terms of allometry, no? And what are the vital cycles of a bacteria, how it reproduces, no? So to me this probably is very important, but I think we need to keep very much in mind, no? How different species age, so that's very, in terms of their metabolic processor, no? Just
we did, so I didn't mention because I wanted to keep the talk short, but of course you are totally right? I have been lying to you. When I say we studied longevity, actually we didn't. We did an allometric correction for longevity. And we use actually, we have used so far in what we have already published, we were using longevity quotient, which takes into account adult body mass, and which is a fair correction. There's literature by George Church and others that show that this is perhaps everything that you need to do, right? Because with body mass, you already take into account metabolic rates and so on and so forth. But what we are doing now is that we have run PCA. We got and we are using the residuals of correcting longevity after taking into account metabolic rate, adult body mass, and a few other correlated variables. Yes, this is totally right. You have to get this right, and we try to. But this is a very relevant point. Yeah, thank you.
Okay. Yeah.
Thank you very much. All right, thank you very much. That was a great talk. Please, my first question has to do with the EGA. I want to know how easy can one access the EGA? And is it for research institutions, or maybe a country has to subscribe to it before they can access it or not?
Okay,
that's the first question.
The European Genome Phenome Archive, I don't have the slides here, but it's an interesting concept. It is free for institutions so far, so you don't need to subscribe. What you need to do is to prove that you are a legitimate researcher that comes from a legitimate institution. And actually, what we take care of is that when a bunch of researchers generated a data set with, say, 10,000 donors, these researchers had to be monitored by an ethical committee. These researchers had to accept some informed consents. There's a lawyer or a lawyer team in the institution that takes responsibility about these researchers behaving okay. So this is a series of conditions that you have to do, you have to accept, in order for this research to be possible. In order to start that data gathering, your data gathering protocol needs to be approved, right? Now, this has happened, let's imagine in Japan. And there's a data set that has arrived to the EGA. Together with this data set, all these informed consents appear, all these ethical requirements and protocols appear. And so, when you, as a researcher from, Barcelona or from Buenos Aires, wants to accept access to this data set, you have to abide by the same conditions that the researchers that originally generated the data set abided to, right? And indeed, the original, we have like more than 2,000 data access committees because the original institution that generated the data has the final say. If they are convinced that you are a legitimate researcher that comes from a legitimate institution, that you have all the lawyers. I mean, a typical thing is that small institutions, for instance, send to us, we have a document that needs to be signed by the legal representative of the institution. And another document that needs to be signed by the IT responsible of the institution. And sometimes we get the two documents signed by the same person. This usually rings, perhaps you are in a small institution. We are not confident that you will have everything that you need. This is how it works, right? So countries do not need to subscribe. It's a question of institutions. And for the individual researchers, we do it for free.
Thank you very much. Please, my second question also has to do with, you spoke about the importance of gathering information. And I understood that sometimes you gather the information, but it might not be important to you at that stage, but maybe as time goes on, you will see the importance of it. So I want to understand, looking at maybe let's say Africa, and then maybe other part of the world, there is less technological know-how in Africa. So how, if they are not able to take some of this data and then store in the cloud or maybe something, and then in future, maybe you only have information of other parts of the world. I'm not sure if that's going to be a limitation, maybe in the study of...
Yeah. So there's, I was saying that you are providing sophisticated questions. This is the most sophisticated of them all because this has enormous sociological implications, and we all kind of notice this, but it also has very important scientific implications, right? So let me say three things. I could say more, but at some point we'll have to go for lunch, right? First thing, there has been a bias, an enormous bias in human genomics. That's because most human data comes from people of European ancestry. And that's it. And that's, of course, a problem, right? Because where you have more genetic variability? In Africa. And actually, I would pose, where do you have more environmental variability? In Africa. So perhaps we are looking at places where the scientific information that we need is not available. But of course, those were the rich countries, they could pay for it. That's one thing. So this bias needs to be corrected. Second thing, there's institutions such as the Global Alliance for Genomic and Health, in which, of course, there are institutions from African countries that are members that are trying to push for this to be solved. And actually, well, I'm a member of the Global Alliance for Genomic and Health. I have never worked in the working groups that have to do with capacitation and data gathering in Africa, but they exist, and that's something that is extremely relevant, right? So this is something that the world has become, second point, this is something, the point you make, something that the world has become aware of, and they are trying to solve it. And for instance, Nikki Calder, she's a great scientist from South Africa. She's spearheading the African Working Group, and she's doing a great, great job, amazing. And my final point is, I feel that this is not only the responsibility of African researchers and of the rest of the world. This is also the responsibility of us as citizens. And I have 2 points about this. First, the selfish point, and second, I would say the unselfish world. First, as a selfish citizen who wants his care to be taken care of, I'm in a hurry for us to get African information, because that's going to be worth tons, much more relevant, for instance, than the information, I don't know, in uniform countries such as Finland, right? Sorry if there's a Finn person in the room. And second, the first thing that we need to do is to make sure that it is African people who do the job, right? And I invite you to donate to one workshop we are organizing in a couple of months from now. We are doing something that is led by Karim Lekadir, who is a fantastic researcher on artificial intelligence based in the University of Barcelona. He has organized a system so that we are going to have a student meeting in Marrakesh, and there are 2 conditions to participate in this meeting, and there's one thing that is an advantage. The conditions are, you get to present your poster only if you are a student who is based in Africa and has an African supervisor. And so we don't want people who, are working in New York and already have a research group there. No, we need to foster that. And second, the advantage is that if you participate in that, we have already like more than 200 applications, right? We will select the 20 most important, perhaps only 10, depending on the cost, posters, and we will try to fund these research projects becoming papers, right, in the African institutions. It's initiatives like this that bring actually the power there that are what we need, also for selfish reasons. And I'm noticing I'm preaching now, so I should stop.
Thank you.
Thank you. Please.
This EGA archive has only human data, or has also data from other species?
It has only human data, and it's data that would allow identifying citizens. If your data has been consented for you to put on Facebook, we cannot have it in the EGA. And the reason is that it's very expensive to maintain the security, right? It's very expensive. And now, you guys, you don't go to your computers and try to hack into the EGA, right? So we cannot accept data that has not restrictions because it would be a waste of resources.
And also for the type of research that is done here at the ICM, the same way that you, for example, were comparing different mammalian species with, let's say, different types of bone density, I can, I don't know, imagine, for example, different marine vertebrates or invertebrates that, I don't know, they can have different tolerance to thermal stress, for example. Would be the same principle that could be applied here, right?
That's right, totally
Okay.
right.
And one last thing, transcriptomic data, for example, you can see it as also phenotypic data, right? Where you know that there is nowadays this view that says that some of the phenotypic variation cannot be explained by genetic variation and is explained by the interface of epigenetics. No, how would you put epigenetics into this big frame?
Well, actually, we are indeed putting epigenetics into this frame, right? I didn't explain this, but so one has to think that in many senses, epigenetics or transcriptomics are phenotypes, right? One result that sometimes shocks people is that when you look at the epigenetics of different tissues, they change. And there's a larger change between the epigenetics and the transcriptomics of heart and I don't know, gut, than between human and gorilla, right? Actually, think about it, right? If you get two hearts, boom, 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 you see they are two hearts. If you get a heart and a piece of gut hanging, you see that they are a heart and a piece of gut. And then they ask you, what species? And you go, no idea, right? So this is relevant. In a sense, we have a bunch of epigenetic and transcriptomic data from these same primates, and we can run analysis, genome phenome analysis, using these as phenotypes. However, we don't need to limit ourselves to that, and we can do what you are suggesting. We can use these two kinds of information, epigenetic variation, and see how if we have information about phenotypic diversity as well, how these influence the phenotypes together in interaction with the genomic variation data that we have. So yes, this is certainly a possibility, and eventually the community will get to it.
All right, thank you.
Okay.
Any more questions? No? All right, so thanks very much again, Arcadi.
My pleasure.
Pleasure.